Welcome to Unit 4, Lesson 2, Part 3. So we're going to pick up and talk about lysosomes. And if you recall, when I was talking about vacuoles in animals, I said they are similar in size to lysosomes and vesicles. So we're going to choose a similar object here. And we're going to call this one a lysosome. And for you to be able to tell that that was a lysosome, I'd have to give you additional information. I could tell you that it was acidic, which means it would have what pH? High or low? That's correct, low. Or I could tell you it had or contains something called hydrolytic enzymes. Those two things are a dead giveaway that you're talking about lysosomes. In a second, hopefully you get the pun I had in there. It wasn't intentional, but it is kind of funny. Okay, so we're going to talk about their structure. At first, as I said, they sound pretty much like vacuoles. The lysosome is a rather interesting organelle. There are some there's at least 40 diseases related to them, and there's sort of cool things that they can do. So structurally, they are a small, membranous sac. Typically larger than vesicles. But smaller than mitochondria. On occasion they can be quite large, but generally if they're abnormally large, that's an indication that they're defective. And they contain acid. In fact, they have a pH of 4.8. And your cell is just slightly basic, just over a pH of 7. So this is pretty low for it. And as I said above, hydrolytic enzymes. These are enzymes that perform blank reactions to blank proteins, carbs, and nucleic acids. So because they're called hydrolytic enzymes, which type of reaction do you think they perform? That's good if you said hydrolysis. And the general class of a hydrolysis reaction is a degradation. So they perform a hydrolysis reaction to degrade, or the more everyday expression, break down proteins, carbohydrates, and nucleic acids. Okay. So here, structure flows right into function. Some of the other functions, so we'll start here. there's kind of an overlap between the function and the structure. They are used to digest engulfed particles. So they can fuse with endocytic vesicles that are bringing in the particles from outside of the cell. Such as nutrients, but also bad guys, bacteria, and even other damaged cells in your body. Now, the whole purpose of the lysosome is to keep the hydrolytic enzymes away from the other parts of your cell, because the hydrolytic enzymes are very dangerous to the cell itself, because the very things they digest are what also make up your cell. So they have to keep it away from the cell content so they don't start breaking down the cell itself. And thus are sequestered within the lysosome and are only active within its low pH. If they get out into the cytosol, it has a much higher pH, just above 7, so they would no longer work. Because if you remember from our previous units, at a higher pH, the hydroxide ions will do what to the enzyme? The enzymes become, what's that word when you destroy its function by destroying its structure? Fancy word, starts with a D. Good, denatured. These guys are really fun. I don't know if fun's the right word, but they get a really cool nickname. Okay. Nicknamed the suicide sack. Because if a cell is severely damaged, its lysosomes will release their contents en masse, ensuring the cell's destruction and digestion. And the other interesting thing is there are numerous lysosomal storage diseases. So there's about 40 hydrolytic enzymes, different enzymes that are found inside the lysosome, and when one of those enzymes doesn't work, something won't get broken down properly and it will build up inside the lysosome and cause a variety of diseases. And one example is a devastating disease called Tay-Sachs disease. And unfortunately, this leads to death in early childhood. And it is a genetic disease. It's recessive, so generally both parents have to carry a defect in the gene that codes for the enzyme that leads to this. And in certain populations, they test for it amongst couples because it's known that the mutation is higher in those populations. But every so often, two people get together, have no idea they're carrying this mutation, and then their child has the disease. And that's where the case of genetic counseling comes in, where if they decide to have further children, they will go through a genetic counselor and sort of look at their options for protecting that child. So if we're going to talk about, back to our analogy, this one's a little trickier, okay? If the cell is like a car factory, then the lysosome is like, now what makes this tricky is you need to have something occurring in the car factory which is dangerous but necessary to break things down. And the best thing I could come up with is then the lysosome is like a paint, because you got to paint your cars. So paint or chemical, because I'm sure they use a variety of chemicals in the process. Recycling room. Within the factory. Where through dangerous chemical processes the materials, being the paint and the chemicals, are broken down and turned into reusable substances. So I think that gets the idea across that you're taking something and you're putting it in a place where it will be protected and safe. You're doing a dangerous reaction, but what you get out of that reaction is useful. So the next organelle we're going to talk about are peroxisomes. And again, peroxisomes structurally look very similar to lysosomes and vacuoles. So we'll pick this guy over here. And for you to understand that that is a peroxisome in a picture, we would have to tell you either that it contains hydrogen peroxide, that's where the name comes from, which has a chemical formula of H2O2, and enzymes called catalases. We're actually going to play with catalases at the end of the year when we do our experimental design unit. It's ideal for studying how enzymes function. Okay? Structurally, as I said, they look quite similar. To lysosomes and vacuoles. They are small membranous sacs that are comparable to lysosomes and animal vacuoles in size. Where they differ is they contain hydrogen peroxide. Yes, we are talking about that stuff that typically comes in a little brown bottle and some of you put on your infections. Not a good plan, by the way, because it kills your cells, not just the bacteria infection. And catalases. Which are also known, aka peroxidases. That break down hydrogen peroxide. So they don't contain hydrogen peroxide because we want them to contain hydrogen peroxide. They contain that as a byproduct of the reactions they perform, and then we get rid of it using catalase. Okay? And they are abundant in kidney and liver cells. Functionally, they carry out reactions that produce the dangerous byproduct H2O2. So we don't, as I said, we don't want hydrogen peroxide in our cells, but when we are carrying out reactions, H2O2 is produced. Interestingly, there are some recent suggestions that we do need some level of H2O2 in our cells, but it has to be tightly regulated, and that's what the peroxisomes can do. So, what type of reactions produce H2O2? Those involved in synthesis of bile acids or bile salts. This is why they're found in liver, because one of the roles of the liver cells is to make bile. And degradation of lipids. So when you're breaking down lipids, you produce hydrogen peroxide as a byproduct. And detoxification of alcohol. And I know none of you would ever drink, especially at your age, but if you do drink a lot, you will notice in your liver an increase in the amount of peroxisomes in the cells. So the peroxisomes cause or carry out these reactions, produce H2O2, which can adversely affect cells. So then it promptly gets rid of the H2O2. So then convert H2O2 into harmless and useful products. And the reaction is, and we'll be looking at this, as I say, in our experimental unit, H2O2, that's hydrogen peroxide, goes to H2O, what's that? Good, water, plus O2, oxygen gas. Both of which are things that are helpful to our body, not harmful. And the way it does this is using the enzyme. Back to my obsessive use of green for enzymes. If the computer cooperates, there we go. Catalase. Okay. So, or in some cases they're called peroxidases. Catalase is just a specific example that we'll be using in our lab. And to look at our functional analogy, so the cell, if the cell is like a car factory, then, okay, now we have to think about something that occurs in the car factory that produces a dangerous byproduct that we clean up before we get out of, it gets out of the factory. And the best thing I could think of is then peroxisomes. You might come up with a better one. Peroxisomes are like the factory smelter. Some of you don't know what a smelter is. Okay? It's where you heat up and melt various metals to make steel. So, where steel is produced, along with the undesirable byproducts such as smoke. But before that smoke can get out and contaminate either the factory or the environment, the smoke is then cleaned by filters in the room and chimneys before it can escape. to harm the factory or the environment. So the smelter room or building would be like the peroxisome, and the filter would equal the peroxidases / catalase. Okay, so now we're going to take a look at the mitochondria. And we've actually talked about the mitochondria before when we were talking about ATP. I'm going to go into a whole lot of detail, but it has been mentioned before. And if we're going to look at it in the diagram, mitochondria are really easy to recognize in most cell drawings. They're drawn kidney bean shaped, and they usually have a squiggly membrane inside of them. Here's a closer picture, and it has an outer membrane and an inner membrane, and hopefully from the first lesson you remember why there's two membranes. And this inner membrane has folds in it, folds back and forth, and those folds are called, and it's hard to see here, cristae, but I'll write it down below. And inside that inner membrane is a fluid-filled space called the matrix. And I remember the matrix because the matrix is in the middle of the mito. So all them's to go together. Middle, mito equals matrix. And mitochondria are actually one of the easier things to recognize in an electron micrograph, because again, they have very clear shape. So as I said, really obvious kidney bean shape with a squiggly membrane throughout. Those squiggly membranes are really the cristae of the inner membrane. Okay. So we'll do structure first. And as we're quickly learning, structure always relates to the function. So mitochondria. Uh-oh. Pen was doing weird things there. Mitochondria structure. Kidney bean shaped, or jelly bean if that makes you happier. Usually ranging in size from 2 to 5 micrometers. And we're going to look at our composite cell being approximately 100 micrometers across, so it's about 1/20th the diameter of the cell. But remember, cells range vastly in size, so we're just going to say that's the size of our composite. Our imaginary hypothetical cell. Okay, so it's kidney bean shaped. It contains double membrane. So if each membrane is a lipid bilayer and it's a double membrane, it will have 4 lipid layers and own DNA, which is supportive of what theory? When we're talking about the origin of mitochondria? That's right, supportive of the I'm helping you out here, biotic theory. The one that says the poor little aerobic prokaryotic cell was engulfed by the larger prokaryotic cell, but actually it wasn't that poor off because it didn't eat it in the end, and they survived together to live happily ever after to make a eukaryotic cell. So we look at them functionally. Okay, they convert glucose to what molecule? Yes, ATP, which serves as the blank currency of the cell. That's correct. Energy. Now, important to it doing this, we have to go back up here, is the cristae. And the cristae have many folds, or are many folds, equals many folds in inner membrane. Why do you think we'd have all these folds in the inner membrane? What do they do to the overall amount of membrane? That's right, they increase surface area, which increases ability to produce ATP, because it's actually quite a complex process to produce ATP, and many of the enzymes are located on the cristae membrane. And the more membrane you can have, the more enzymes you can have, the more enzymes you can have, the more ATP you can produce. Okay, so that is the function. Now, if we were to take it to a car factory analogy, okay, so if the cell, I'm going to try and squeeze it in on this page, if the cell is like a car factory, then the mitochondria, and we're just going to shorten it to mito, the mitochondria are like electrical transformers, and you can see these outside of your house. They're the big gray boxes that are up on the electrical poles. And they might be in some places inside factories, they might be on the outside just as electricity is coming in, but they are like electrical transformers that step down the high voltages, and electricity is sent by high voltage because it's more efficient, to transport it that way. To lower voltages that are safer and easier to use. kind of similar to the idea of when we're converting glucose, which can store a fair amount of energy, fairly efficient, down to ATP because it's easier and less wasteful to use. Okay. Alright, we're going to take one more break before we finish this off, so we'll do it in 4 parts total. If you have any questions about that part, you've been watching this at home, you're not sure what I'm talking about, please come see me. And also I recommend you go to the website Cells Alive. It's a very nice website. It's one of the best I've seen. So Cells Alive. And you can sort of have an interactive look at the various organelles. Have a good day, and I will see you in class.